In this video here, we are going to look at chapter 6.2 in the IGCSE Biology book, which mainly covers section 7.4 in the syllabus. As always, the videos here are made for my class, but if anyone else can use them, you're more than welcome, and any errors here are mine and mine alone. So 17.4 in the syllabus actually covers a lot of ground, and as always, the vocabulary is big, so it's a good idea to practice it. We need to look at inheritance as transformation of genetic information. We need to be able to look at genotype and phenotype and describe the difference. We need to be able to look at homozygous and heterozygous. We need to look at dominant alleles and recessive alleles. We need to be able to look at diagrams and use Punnett squares. And we need to be able to use genetic diagrams to predict result of a monohybrid crossing. So let's have a look at this. Not in this order, but we'll have a look at it. Let's go. It's important that we know what the difference is between genes and alleles. A gene is something on our DNA that codes for a specific protein. Alleles are different variants of that gene for a trait. And you can have different versions of them, then you're a heterozygote, or you can have the same version, and then you're a homozygote for that allele. We also need to be able to distinguish between genotype and phenotype. And the example I use here is for a butterfly. And if you have big A, you're homozygote. If you have small a, you're homozygote. And if you have small a, big A, you're heterozygote. Now, the genotype big A will give the phenotype of dark wings, and the genotype of small a will give a light phenotype. But the genotype of small a, big A will still be dark, which means that you can have different genotypes giving the same phenotype. As we saw in the earlier example, if you have small a, you will be light, but if you only had just one of the big A's, that will determine the color. That is what we talk about in genetics as being dominant and recessive alleles. A dominant allele is one, if it's just there once, it will determine the phenotype. While if you have the recessive, you need both of them for them to be able to determine the phenotype. An example here can be for blue eyes. You have one parent here who is homozygote with a small b, and one that's heterozygote with big B, small b. And here we can see that they can get both offspring, but you only get the blue eyes if you have the small b genotype. So the genotype again determines the phenotype, and the dominant allele will be dominant. So if it is there, it will determine the phenotype. But you can still have a genotype where you carry the recessive allele without it being expressed in your phenotype. Another option is what's called codominance, where both the genotypes go in and have an effect on the phenotype. In this case here with flowers, if you have the big R, you'll get a very red color. And if you have the small r, you'll get a white. But if you have both the big R and the small r, well, then they both have an effect on the phenotype. So in codominance, both of the alleles will have an effect on the phenotype. We'll see how the flower will look. So another very important keyword here is haploid and diploid. Most of the cells in the body here are diploid, which means there are two sets of chromosomes. It's a pair of chromosomes. But when we do reproduction, we need a haploid cell with only one set of chromosomes. So you can get one set from the father and one set from the mother, and those will then combine. This is important when we do our crossings, our Punnett squares, and I'll show later that remember the parent might be diploid, but the sex cells it sends to form the next generation are haploid. So here is an example on how to use this. It's called a punett square, and it's being used a lot in biology. It goes all the way back to our old hero, Gregory Mendel. But the idea is that if we want to see what the different outcomes are, we put up the genotype of each parent, and then we make haploid cells. Because remember, the parent, in this case here, might carry both the big G and the small g, but since it only passes on one allele to its offspring, it can either pass on the big G or the small g. In this case here, we have one parent who is a homozygote with recessive alleles, and we have one who's heterozygote with a dominant and a recessive allele. In this case, if we want to see what are the possible phenotypes of our offspring, we do a crossing here, and we put in the possible new genotypes in each square, and we'll see there is like two of the outcomes will come big G, small g, which will give us the green color, and two of them will be small g, which will give us the yellow color. If ever there is an exam question where they ask you about doing crossings, please draw a punett square. It's a good way to show you know what you're doing, and it's a good way to get the points. Last thing we're going to look at here are probabilities in genetics, and whenever you do that, please always, if you can, make a punett square. Here again, we are looking at two heterozygote individuals, both having the small a and the big A, which means if you look at their phenotype, they will both look like the big A phenotype, as we assume here that big A is the dominant allele. So what are the odds of them having a offspring which is homozygote, recessive, and thereby show the different phenotype? Well, in this case here, we have four possible outcomes. We have big A, that's 25%. We can have a big A, that is a 50%. And we can have a small a, which is again 25%. So the odds, or the chance, or the risk, depending on how you want to see it, of us giving the small a genotype and thereby expressing the small a phenotype will be 25%.